Good morning. Hello, everyone. Today, I'm really honored to have this opportunity to introduce my study and this conference, and many thanks for the invitation. And my name is Chen Gong from the Tsinghua University School of Basic Medicine. And my study mainly focuses on the roles of the gut commensal bacteria in the regulation of the vector competence of the mosquito to this arboviruses infection. So here, we identify the naturally isolated symbiotic bacterium can suppress this Flavivirus replication infection in the mosquito vectors, therefore suppressing the dissemination or distribution of the mosquito-borne arboviral diseases in nature. Firstly, I want to introduce this background of our study. And as we know, this mosquito serves as a primary vector for many human virus dissemination and transmission in nature, in which Aedes mosquito and Culex mosquito are major vectors for many mosquito-borne virus transmission. In nature, infection of the mosquito-borne viruses can cause severe clinical consequences such as encephalitis, meningitis, or hemorrhagic fever. The mosquito-borne arboviruses actually belong to the 3 independent virus families, including the Flavivirus family, Togaviridae family, and the Buniaviridae family in the world. And in the past 20 years, many mosquito-borne arboviruses, such as West Nile virus, Chikungunya virus, Zika virus, or dengue virus, widely spread in the world and causing the major concern of the public health. For example, the dengue virus is considered as the most prevalent of the mosquito-borne arboviruses emerging or re-emerging in more than 100 countries worldwide. And so every year, there are hundreds of the millions of the people got infected by dengue and other mosquito-borne arboviruses in the tropical area, and millions of the patients need to be hospitalized and resulting in the 10,000 of the deaths every year. So this mosquito-borne arboviral diseases is considered as a major concern of the public health. As we know, this mosquito-borne arboviruses maintain their life cycle between the host and the mosquito vector. In this process, firstly, mosquito got infected. Then the infected mosquito can transmit these virus particles to the human or another water-breathing host. Then the virus can very fast replicate in the host blood and develops very high viremia in the blood circulation system. During the viremia period, a clean mosquito can further acquire this virus, and from the infected host, the virus goes back to a new mosquito body, infect mosquito cells, and enable this mosquito has capability to spread this virus to another host through the next mosquito biting, complete this transmission life cycle. In this transmission life cycle, actually, this arbovirus acquisition process from infected host back to a feed mosquito vector serves as essential steps in this virus life cycle. However, quite a few studies touch on this molecular mechanism of this arbovirus acquisition by the blood meal. So the key factor to determine the effectiveness of this arbovirus life cycle is still largely unknown. And we just mentioned that these Flavivirus particles can be acquired into the mosquito gut lumen from the blood of the ceramic host. And so in this process, the Flavivirus particles can first infect its epithelial cells of the mosquito guts, disseminate into the mosquito hemicone, infect the salic gland, and enable this mosquito has capability to further spread virus to another host, so next mosquito biting. And so this mosquito guts and enriching the community of the microorganisms serve as a primary viral entry site to initialize this virus infection. So gut microbiota maintaining the homeostasis and in mosquito actually largely regulates this Flavivirus replication, infection, and in the mosquito vectors. Indeed, our pre-studies have focused on the roles of the gut commensal bacteria in the regulation of the Flavivirus replication infection in the mosquito vector. In the pre-studies, we first used this lab-adapted this Aedes aegypti strain, and for the investigation, we first identified the Syringium macensis as gut commensal bacteria to promote this Flavivirus infection in mosquito by secretion of the bacteria factor named as SS mihancin. The mechanism study suggests that this SS mihancin can directly digest this polysaccharide layer of the mucin protein, disrupt this physical barrier on the mosquito gut epithelial cells, therefore facilitating the Flavivirus replication infection in the mosquito gut epithelial cells. And so actually, these studies, and we firstly identify the gut commensal bacteria can determine the vector competence of the mosquito to the Flavivirus infection, the data published in the Cell Host Microbe and in 2019. However, and in the upper mentioned study, and we just identified the Symbiotic Bacterium, this bacteria, and from this the lab adapted this Aedes aegypti Leucophila strain. So it's very difficult to establish a correlation between the gut residence of the Symbiotic Bacterium in the guts of the field mosquito and the prevalence of the dengue in nature. So in the next experiments, and we want to isolate the cattle gut commensal bacteria and from the field mosquito, and further investigate, identify this key gut commensal bacteria to determine the vector competence of the field mosquito and in nature. So first, we want to introduce our study from October in the 2020. As we know, the Yunnan province and serve as a major area for the high prevalence of the mosquito-borne Flavivirus diseases in China. And so we first established this expert station of the Yunnan province with my name, called by the Chenggong expert station of the Yunnan province. Yeah. So, and in basis of this expert station, and actually we collected is 10,000 of the field Aedes albopictus mosquito in the border area of the Yunnan province, and isolated the gut commensal bacteria for the further investigation. As we know, quite few these Aedes aegypti mosquito are distributed in Yunnan province. So the Aedes albopictus mosquito serve as a primary vector for the dengue endemic in Yunnan. So we first collected this 10,000 of this field Aedes albopictus mosquito from the Yunnan province, and so we isolated the gut commensal bacteria for the further investigation. Finally, another 55 the bacterial species has been identified from the middle guts of the field Aedes albopictus mosquito from the Yunnan province. We next want to further investigate the roles of this gut commensal bacteria in the regulation of the dengue virus infection in the Aedes albopictus mosquito. And in all tested is the gut commensal bacteria. One bacteria and belong to the Rosenbergiella genus largely suppressed under this dengue effectivity and the prevalence in this Aedes albopictus mosquito. And so suggesting this bacteria is a targeted bacteria and for our study. And so based on the previous literature, we learned that all the bacteria belong to the Rosenbergiella genus and has been identified as plant nectar and associated with the microbial community. So we named this bacteria, it's a new species, as the Rosenbergiella 146 for the further investigation. To further investigate the roles of the Rosenbergiella 146 in the dengue virus infection in mosquito, we next and already introduced this bacteria into a gut lumen of this antibiotic-treated mosquito. Subsequently, and these mosquitoes was already infected by the blood meal and sacrificed at 8 days post this infection. The data suggesting the prevalence of the dengue and Zika infection can be largely reduced by the gut residence of this Rosenbergiella 146 in both its Aedes aegypti and Aedes albopictus mosquitoes, suggesting the gut residence of the Rosenbergiella 146 can largely reduce its vector competence or susceptibility of this blood-feed mosquito to a dengue or another Flavivirus infection. In the first section, and our data suggests that this Rosenbergiella 146 is a gut commensal bacteria and reduce under this vector competence of this blood-feed mosquito to dengue. In the next section, we will continue to introduce this mechanism and what happens. And so here, we identify the Rosenbergiella 146 actually can secrete this bacterial factor to acidify the mosquito gut lumen and so to inactivate the virus particles acquired from blood of the ceramic host. And as we know, this bacteria can use many factors to interact with the host cells for their successful survival, and including the bacteria, cellular components, secret protein, and metabolic compounds. And also, to further identify which bacterial factor suppresses the Flavivirus infection in the mosquito vector, we next incubated this bacteria and for the 72 hours at 30 degrees. And subsequently, this culture supernatant will be collected by the centrifugation and filtration. However, this bacteriolysis will be generated by the sonication. Either this the culture supernatant or the bacteriolysis will mix with the human blood and the dengue supernatant for infection of these antibiotic-treated mosquitoes. The data suggesting that actually the oral introduction of this culture supernatant, but not this bacteriolysis, resulting in the reduction of the dengue virus infection in both these Aedes mosquitoes, suggesting this extracellular effector and this culture supernatant secreted by the Rosenbergiella 146 and responsible for the high resistance of the blood-feed mosquito to dengue. And indeed, there are three possibilities, and for this extracellular effect, is a cultural supernatant suppressing this flavivirus infection, including these small peptides, the secret protein, and the metabolic compounds. And so we next want to further identify this bacterial effector suppressing this flavivirus infection, this cultural supernatant, by the further fermentation and functional study. In this study, we first separated this cultural supernatant by the 3 kD, the centrifugal filter. In this system, these upper retentive liquids include protein. However, these lower filtrate liquids include small peptides and the metabolic compounds. Either these upper protein retentatives or the lower filtrate liquids will mix with the human blood, dengue supernatants, for infection of these antibiotic-treated mosquitoes. The data suggesting the ingestion of these upper protein retentatives, but not these lower filtrate liquids, can largely suppress this dengue virus infection in both these Aedes mosquitoes, suggesting this bacterial effector and in this cultural supernatants may be the protein secreted by the Rosenbergiella 146. Subsequently, these upper protein retentatives will be separated by the SDSP gel and identified by the mass spec. So the secret protein with the top 10 high score in the mass spec will be selected for expression and purification in the E. coli expression system. So 10 micrograms of the purified protein mixed with the human blood, dengue supernatants, for infection of these antibiotic-treated mosquitoes. The data suggesting among all these tested proteins, ingestion of the recombinant purified glucose dehydrogenase can significantly reduce this dengue infection in both these Aedes mosquitoes, suggesting this protein is a targeted bacterial factor suppressing this flavivirus infection. We name this protein as the Rosenbergiella glucose dehydrogenase and for GDH for the further investigation. So we next want to further investigate the roles of the GDH in the dengue virus infection of the mosquito. As we know, the glucose dehydrogenase can effectively convert this glucose to gluconic acids. A large production of the gluconic acids by the bacteria actually can effectively acidify the surrounding environments. So we next want to use this artificial blood mimicking medium and containing the pH indicator and to assess its acidifying capacity of the Rosenbergiella GDH in the mosquito gut lumen. And in this study, we first feed this artificial blood mimicking medium and supplemented with a pH indicator to the Aedes albopictus mosquito. After either this 50 or 30 minutes post this blood meal, the guts of this feed mosquito will be collected to determine the pH value. And the data suggesting the medium can be rapidly and actually acidified to the pH 6 and in this Rosenbergiella 146 and colonize this mosquito guts, but not in this GDH knockout and this the bacteria colonize this guts, suggesting the GDH and serve as its played an essential role in the acidification of the mosquito gut lumen. So we next want to further assess is the acidifying capacity of the Rosenbergiella GDH in the guts of the mosquito feed this human blood, consistent with the results by feeding this artificial blood mimicking the medium. We can see that colonization of this Rosenbergiella 146 can effectively acidify the gut lumen to the pH 6 to 6.5 in mosquito. All this data suggesting this Rosenbergiella 146 actually can acidify the mosquito gut lumen by secretion of the bacterial factor named as the GDH. And so the next scientific question is how the GDH-mediated acidification of the mosquito gut lumen can suppress this flavivirus replication effect in the mosquito vector, right? So the well-known phenomenon is the envelope protein of the Flavivirus variant can undergo irreversible structure change at the acidic condition and with this pH value below the 6.5, which naturally occur during the virus entry into the endosome of the host cell. Indeed, the structure change of the E protein and can lead to a complete loss of this virus effectivity. And so the pH value and below the 6.5 in the surrounding environment indeed and mimic the acidic condition in the endosome of the host cell. This data suggests that, and actually this GDH-mediated acidification of the mosquito gut lumen can directly inactivate and these virus particles acquired from blood of the ramic host, and therefore suppressing this virus replication effect in the mosquito vector. Up to now, we learned that it's a Rosenbergiella 146 and it's environmental bacteria. and gut residence of the Rosenbergiella 146 and can largely reduce its vector competence of the field mosquito and to dengue. So the last section, we want to further investigate whether this semi-field intervention with this Rosenbergiella 146 may reduce this dengue prevalence in the field mosquito and from the dengue endemic area such as Sichuan-Banna in China. And so first we want to investigate whether this Rosenbergiella 146 in the mosquito breeding water and can be effectively transferred into the guts of the emerging adult mosquitoes by the transstadial manner. In this experiment, the serum concentration of the Rosenbergiella 146 was initially added and into the water with the eggs of the field Aedes Mosquitoes mosquito, data suggesting the condensation of this Rosenbergiella 146 can be abundantly detected and in the guts of this the emerging adult mosquitoes, larva and pupa and the mosquito and adult mosquito. And subsequently, we want to further investigate and the capability and of these mosquitoes and hatch and from the water with or without this Rosenbergiella 146 intervention, the dengue virus acquisition process. In this experiment, actually, the mosquitoes and actually was well, we hatched from the water and with this Rosenbergiella 146 intervention. And subsequently, these hatched mosquitoes were allowed to feed on this dengue-effective viremic host, 86 mice, and from 1st days to 5 days post-animal infection. And at 8 days post-blood feeding, we collect mosquitoes, sacrifice mosquitoes to determine the virus burden by the Taichman qPCR. The data suggesting addition of the Rosenbergiella 146 and actually to the mosquito breeding water and can significantly reduce its prevalence of the dengue in the Aedes Mosquitoes mosquito from Sichuan-Banna, suggesting the intervention of the Rosenbergiella 146 in the mosquito breeding water can largely reduce and the dengue vector competence or the susceptibility of the field mosquito from Sichuan-Banna. It's a dengue endemic area in China and to the dengue virus infection. And subsequently, we want to further investigate whether it is a semi-field intervention of the Rosenbergiella 146 in the mosquito breeding nature environment may reduce the dengue effectivity in the field mosquito. So the semi-field studies were performed and within the enclosure named as a mosquito sphere and in the Sichuan-Banna. And to create the nature environment and for the mosquito breeding, the local soil was added and into the water and from the Sichuan-Banna for the mosquito breeding. And in this experimental setting, the serum concentration and of this Rosenbergiella 146 cells and also the eggs of the field Aedes Mosquitoes mosquito from Sichuan-Banna was co-introduced and into the water for the mosquito breeding. And actually, and the Rosenbergiella 146 can effectively colonize and in the guts of this Sichuan-Banna Aedes Mosquitoes mosquito by the transstadial manner. So in as affected under this emerging adult mosquito by the blood meal, the data suggesting the gut residence of the Rosenbergiella 146 significantly reduces the dengue prevalence and in the Sichuan-Banna Aedes Mosquitoes mosquito. All this data suggests the semi-field intervention of the Rosenbergiella 146 and actually and in the mosquito breeding nature environments such as the mosquito breeding water can largely reduce its vector competence of the field mosquito to dengue, therefore suppressing this virus dissemination or transmission by mosquitoes in nature. So here I want to summarize our study by a simple diagram. In this study, actually we first identify the Rosenbergiella 146 as a environmental bacteria, also a gut commensal bacteria for mosquito, can largely suppress this Flavivirus replication effect in the mosquito vector. And so the mechanism study suggests that actually under this the Rosenbergiella 146 can secrete this bacteria factor and to acidify the mosquito gut lumen. And so the GDH and this bacteria factor mediated acidification of the mosquito gut lumen, directly inactivate these virus particles acquired from the blood of the viremic host, therefore suppressing under this virus replication infection and in the blood-feed mosquito, and therefore reducing the vector competence of the field mosquito in nature. And up to now, we learned that it's a Rosenbergiella 146 is environmental bacteria. Gut residence of the Rosenbergiella 146 can largely reduce this vector competence of the field mosquito to dengue. And so, and for data suggesting this semi-field intervention of the Rosenbergiella 146 and in the mosquito breeding water can effectively suppress and this vector competence of the field mosquito from Sichuan-Banna and it's a dengue endemic area, and to a dengue or not a flavivirus infection. Therefore, and the surprise and this virus dissemination or transmission by mosquitoes in nature. And for this study, actually, we provide this new biocontrol strategy to prevent the dissemination or distribution of the mosquito-borne flavivirus diseases. The data published and in the Science and in this April of this year. And for these studies, I want to give many thanks for my lab member, my collaborator, and also supported by several mega grants from the international and the Chinese government. Thank you.
Well, thank you, Cheng Gong, for his wonderful talk. So time for question.
Thank you. That was a great talk and a great strategy. So I think this has a lot of potential and a lot of promise. My question here is like, did you look at the flora or of the bacteria that would be present in the saliva of those mosquitoes in which you recolonized with this Rosenbergiella to see if that would be also provided in the mosquito bite and also inactivate any residual viruses that could be when the mosquito bites the person or the animal.
Very nice question. Actually, we detected this location of this bacteria in the different mosquito tissue. However, and this bacteria and just and specifically localized and in the mosquito guts is a gut commensal bacteria, but we did not detect this bacteria and in the mosquito cell gland or another tissue. However, it's amazing of note, and this bacteria actually can be transferred or transmitted and by the vertically and from the adult to eggs, and also the horizontally and the mating, and also it can be transmitted, and so Thank you.
Really beautiful work. You need to submit a mini review to Journal of Virology.
In the future, thank you.
But my question is, you always had a few mosquitoes that were that you had treated with the Rosenbergiella. Yeah, he's better. He's better at saying this. That maintained high levels of dengue. Did they not get colonized, or do you not have the pH change, or is there another bacterium there that's out competing?
Yeah.
Do you know what's happening there?
Yeah, I think the colonization of this Rosenbergiella 146 largely depends on the physiological status of the individual mosquito. So we have tested actually the threshold of this bacteria. I mean, the surprise is the flavivirus infection mosquito needs the enough number and of this bacteria. Usually, and we detected is hundreds of the thousands, and around this 500, 1,000 this bacteria in the mosquito guts can enough and to suppress this virus infection. Maybe for some mosquito, and this number of the bacteria is maybe not enough. Maybe a lot of the factor or reason can regulate this the residence of this bacteria in the mosquito gut. Maybe in the future we'll find the more the effector and to regulate and this phenomenon. Thank you very much.
Yeah, thank you. A fantastic study. It's a very different paradigm from sterile insect technique or Wolbachia. So the question is actually a follow-on to what Anna just asked. Do you know anything about, when you were doing the field studies, about the actual parameters of how often you have to add it? Is it maintained? Like how do you continue to use this, let's say, as a viable practical intervention?
Yeah, I think it's a very good question. Actually, and we just we are doing is the larger experiments in the village and in nearby this Xizhuangbanna. We put and set many this bottle is put a lot of water, and maybe in a bottle is just 10 liters water, and we put this bacteria directly into the water, because this water can attract this mosquito lay eggs and into this the bacteria contaminated water. After this adult mosquito emerge from the water, and actually they can carry this bacteria. So we put in one village, and actually we put maybe the 200 or the 300 and this big cage, and actually to disseminate this bacteria in nature. Yeah, that's is current this approach and for the study. Yeah.
Okay, you first.
It's a fantastic talk. And the one question is, right now it's basically a theory, it's basically low pH is inactivated the dengue virus. Have you able to try to do serial passage to get somehow the pH resistant to the virus to put into the mosquito to say if it's a pH is the only thing functioning, maybe some other mechanism involved from the bacteria to inactivate the virus?
Yeah, I think this structure change and the pH mediated structure change of the Flavivirus variant is a physical and it's a phenomenon. Actually, yeah, because it's a virus, use a low pH this condition to encoding and release this genome. So it's I think the virus maybe not will have this another mechanism.
I was just wondering, have you the serial passage, you just gradually reduce the pH to say if you able to find some of the mutation might affect, still in the like pH 6 or maybe below, they still can trigger, but they are what you call the inactivation of the pH will be a little bit lower to look at to compare it.
Yeah, I saw some of the viruses may maintain their tolerance and in the around pH 6.5 or 6.4. But if you reduce this pH value to the 6 or 6.1, all viruses will be killed. Maybe I will try this method in the future. Yeah, thank you.
Dear Professor Cheng, I have technical questions.
Okay.
So you just mentioned 55 types of microbiota were isolated. So for human beings, the numbers should be more than that.
Sure.
So what is the key step to narrow down the size and to focus on the specific type of microbiota for future research?
Yeah, I think this is a very good question.
Okay.
Because for the human microbiota, it's much more complicated, right? And so actually we isolated this bacteria from mosquito guts. Most of this, not most, maybe a lot of the mosquito guts commensal bacteria and is from the environment. So a lot of the mosquito gut bacteria is and actually need oxygen. Right. So it's culturable. But for the human gut commensal bacteria, it's very difficult and for the culture, and maybe it's a difference. Yeah.
Okay, thanks.
Thank you.
Because of the time limitation, we have to move to the next speaker. Thank you.
Okay.
Thank you.
Thank you.